your syllabus. Small little part is left to be discussed. Last time we have discussed about linkage and crossing over. That is, how are genes linked or they are not linked, and if genes are linked, then when they are passed on into the next generation, linked genes in the pair form only get inherited. That means the chances of those two linked genes separating from each other and forming a new combination, that percentage is very low. On the other hand, if genes are not linked, then they are likely to be separated during crossing over and they may give rise to new combination. So their percentage of recombination is high. And this was proved by Morgan when he performed experiments on fruit fly insect, or it is Drosophila melanogaster. Then you have also studied that on the non-homologous region of X and Y chromosomes, there are certain genes present which are responsible for some expression or characters. Now, these non-homologous regions do not have their allele on the other chromosome, because this part being non-homologous, its meaning is the gene on the non-homologous region of X will not have its allele on Y and vice versa. Now we are going to discuss two such genes located on X and Y chromosomes, and they are located on the non-homologous region, and let us see how is their inheritance. Now this inheritance is called criss-cross inheritance, that is mother to son and son to daughter. So this is how the inheritance takes place. And one example is color blindness. Now this color blindness is red and green color blindness, meaning person fails to distinguish between red and green color. So if you have ever seen the application form for a driving license, you will see that there is one question in that questionnaire or in that form, that whether the person applying for driving license is color blind or not. Because if you see in the traffic signal, red and green colors are used. You know, red color is to stop, green color indicates to move on or to drive on, and in such situation, if a colorblind person is driving a vehicle and fails to distinguish between red and green colors of the traffic signal, he or she is likely to be in danger, may meet with an accident. Okay, so that is why this color blindness in day-to-day life has such an important application. Now, let's see how is this color blindness inherited and which are the genes responsible. Now, gene for vision, where we can distinguish between the colors, that gene is present on X chromosome. Okay, which means that X chromosome is present both in males and females. So vision is a character dominant over its recessive form. Now, if recessive gene is present, that is small c is present, then this gene is indicating color blindness. So, gene for color blindness. And now, in the table, I have written variety of combinations. Okay. Now, first understand how are these combinations to be written, and then we will see different crosses. Now, in the first row, I am writing genotypes of male individuals. Now, male individuals, human males, they have X and Y chromosomes. Okay, so that is why X and Y are written denoting basic genotype of males. Okay, then double X chromosomes for female combinations. So every female, we are right now not taking into consideration how is the vision. But when female individual is there, both X chromosomes will be present. So that's why everywhere you will find double X chromosomes. And then afterwards, while writing genotype, now the next thing which comes is whether X chromosome carries capital C gene for normal vision or whether X chromosome carries gene small c for color blindness. And now we will see all the possibilities. Now suppose if male is having normal vision, then X having gene capital C, because capital C is for normal vision. So X capital C Y is the genotype of the male. But if male is color blind, then gene will be recessive, small c, and the genotype of the male individual would be X small c and Y. Okay, so if you understand this basic thing, then writing cross is not going to be difficult. So these are two possibilities in males. So human males will be either having normal vision or colorblind. No third possibility. But now in females, let us see. Females have both X chromosomes in them. Now when there are two X chromosomes, there are three possibilities. Now first possibility is both X chromosomes have normal dominant gene, that is capital C. So when both capital C genes for normal vision are present, then that female is said to be having normal vision. Now, second possibility. Both X chromosomes have gene for color blindness, that is small c. Small c and small c. So on both X chromosomes, the genes for color blindness are present. Then the female is said to be color blind. And the third possibility, she is called a carrier. Now, how is carrier? Carrier is a combination of one normal gene. So, one is capital C for normal vision, and second is going to be gene for color blindness. So, it is a combination. So, this is a heterozygous condition. So, this is how male and female individuals will have different possible combinations as far as their vision is concerned. Now, all of you open your textbook and we will see the two tables given in the textbook on page 63. Quickly open your books. Now, in first cross, you will see that male individual is colorblind and female has a normal vision. Okay? Now, refer this table. You will understand that colorblind male means X small c y, and female has normal vision. So, double X chromosome and both having capital C gene, that is gene for normal vision. Now, obviously, when two parents are going to form gametes, by rule of segregation, you will see that male individual produces two types of gametes because one chromosome is X and other is Y, which are non-identical, and that's why X and Y will separate. So, X chromosome with the gene for color blindness, small c, that goes into one gamete, and Y chromosome goes into the other gamete. Female will give rise to one type of gamete because she has normal vision and both the genes are going to be capital C. Now, when we think of various possible combinations, then from male parent, X chromosome, when fertilizes with one gamete from female, then combination is going to be one normal gene and other is for color blindness. So, if you look at the progeny F1 generation in the textbook, see, first two combinations are having one gene normal and other for color blindness. So, they become carriers. Look here. This combination, one normal gene, other is for color blindness. So they become carriers, and son when born, then sons will have normal vision. So Y chromosome from second gamete when fertilizes with female gamete, now these female gametes, they are having normal vision gene, and that is why son gets normal vision, whereas a female child, that is a daughter who is born, will be a carrier. So here in this cross, what has happened, if you observe carefully, Gene for color blindness was present in father, and from father, daughter receives that gene and not son. Now, let us see second cross. In the second cross, daughter is carrier and male, that is father of the in the cross, is having normal vision. Now let us see what are the possible phenotypes in F1 generation. Now you already know how to write gametes and how to write different possible combinations. So there is going to be normal female child, normal male child, carrier female, and colorblind male. Okay, so there are four possible genotypes in F1 generation in this particular cross, and you will get one normal female, one normal male, one carrier female, and one color blind male. So now if we just study who were the parents and how is the progeny, then you will realize that gene for color blindness was present in mother, and now from mother this gene is passed on to her son. So earlier father transmitted gene to daughter and daughter to her son. Okay, so this pattern is called criss-cross inheritance. Okay, now we will see another example, that is hemophilia. Now in this hemophilia, the medical problem faced by the patient is blood fails to clot if there is an injury, external or internal injury. That is, whenever a blood vessel ruptures. In normal course, what happens? When we get a cut or a wound, blood vessel ruptures. Blood comes out. And when that blood comes in contact with air, then clotting factors, they are triggered and clotting takes place and excessive blood loss is prevented. Now in these patients, patients of hemophilia, blood does not clot, fails to clot, or it might take several hours or a very long time for clotting process, and by that time patient may lose lot of blood. Okay, so that is hemophilia. Now let us see how is this particular hemophilia controlled in human beings and how is the inheritance pattern. hemophilia. Normal gene is capital H, where proper blood clotting takes place, which is present on X chromosome. Its recessive form is denoted by small h. So if small h gene is present on X chromosome, then person is going to show symptoms of hemophilia. Okay? Now again, just like the earlier example, let us write various possible combinations in males and females. Now we will first write genotype in males. Now for your understanding, I am writing X and Y first. Basic genotype of males is X and Y. That's why I have written X and Y genotype. Now in the normal individual, gene on X is going to be capital H. So normal blood clotting takes place. And if the male is hemophilic, then X chromosome will have small h gene, that is gene for hemophilia. Now third case does not exist in males because carrier will be such a person who has both X chromosomes, because carrier meaning one gene normal and other is abnormal. So that possibility does not exist in males. And in females, we will write all the possibilities one by one. But before that, I am writing basic genotype of females. Females have both X chromosomes. So for all the three combinations, I have first written basic X chromosome. Normal will have both genes normal, that is both capital H. Hemophilic means both small h, and carrier will have one normal and one hemophilic gene. Okay? So this is how various genotypes are written. Now we will see in the textbook how is this particular disorder passed on to their children. Hemophilic male marries normal female. Then in that case, if you see the progeny, daughter becomes carrier, and if son is born, is normal. Means when father was hemophilic, gene for hemophilia has passed on to daughter. Now on the next page 64, we will see next part of the cross. Daughter is carrier and male is normal. So here now mother carries gene for hemophilia, and let us see how is the progeny. One normal female, one normal male, one carrier female, and last one is students, there is printing mistake in your textbook. By mistake it is written color blind. Now actually it should be hemophilic person. So make that correction in your textbook. So one daughter, one son normal, one female carrier, and one son is hemophilic. So mother had a gene for hemophilia. She has passed it on to her son. Again crisscross inheritance. Okay. Now study this well. Next time we will see few other disorders.